So, very good morning to all of you. Welcome to the channel Study Ka Enzyme. And here we are going to begin with the first chapter of genetics from grade 12, which I feel is little bit tough for the students. So, I request you all to be here and start learning this. So, let's get started. Okay, so as we can see, um, as we begin with chapter inheritance, principles of inheritance, first I would like to focus your attention on this man. He's Gregor George Mendel. He's the father of genetics. As you can see, he was working on this Pisum sativum. So this question often comes in the examination. What is the organism? What is the plant that he began his trials on? So he worked on Pisum sativum, which is the garden pea, as we have been using it in our houses, and you are much familiar with this also. Okay, so the first thing that we are going to study here is genetics. So what is genetics? What according to you is genetics? So genetics is the study where traits are transmitted from one generation to the other generation. Like, so if we study these inheritance patterns, say we study genetics. For example, you would have seen your mother uh, having eyes like your grandmother, maternal grandmother, right? You might have your nose shape like your father. You might have your hair texture like your mother. So these are all traits which we call characteristics. So where do these traits come from? From the previous generation, from the parents, right? Parents, grandparents. So transmission of these traits is known as genetics, right? So if we study these traits, it's genetics. And if I talk only about the transmission of these traits from one generation to other, that's inheritance. All right, so next we study what are variations. Variations that you can see is the difference that you see among yourself and your sibling. You would have seen no two siblings being similar except those identical twins, right? So you might be looking similar to your brother, your sister, but you're never identical, right? So these differences that exist between the siblings, that exist between the parent and the siblings, so these differences are known as variations. Okay, I have a very nice video image also to share. Let me see if I can show that to you. I have an image where we see a transmission. Like it's a cartoon, but I hope you enjoy looking at it. Just see. So, it's just a prank, like the child having these ears, hair on the ears, just like the father, right? So these are also some of the traits. You would also have seen uh, Amir Khan with long ear, that are also the genes, right? So these genes are transmitted, and the transmission of these traits will be because of the will be because of what? Those genes, correct. Characters. So traits are characters only, okay, that we have always been seeing. Okay, so another thing that children are usually confused about is alleles and allelomorphs. So what are they? See, I have always been telling you that there are two chromosomes in a diploid cell. You must have seen in the previous video also. There are two chromosomes, one from mother, one from father, right? So if I talk about eye color, right? So eye color may not be similar uh, in both the parents, right? Mother might have a different eye color, father might have a different eye color. So if a child inherits both the chromosomes, so he has both the alleles. For example, if I tell you this, let me just a moment, give me a while. If I try showing this to you on this whiteboard, suppose we have two chromosomes. So gene of eye color will be there on both the chromosomes, right? Both the chromosomes if I draw both the chromosomes, suppose this is one chromosome, this is the second chromosome that a child might have inherited from the mother, the other one from the father, right? So if I make eye color here, gene for eye color here, suppose this is from mother and this one is from father, right? This one is from father, this one is from mother. So mother might have blue eye color, take it, father might have black eye color. So this child has one gene, that is eye color gene, in duplets. He also has a gene for blue eye color, and he also has a gene for black eye color. Okay? So both of these are known as alleles. Understood? So, what we can say, different forms of a genes will be alleles. Let me give you more examples. For example, you must have seen people with different hair color. They might have blonde hair, they might have black hair, they might have brown hair. Okay. So, all these colors will be because of the gene for hair color. Okay. And these black, brown, blonde, so these are all alleles. Understood? Let me give you more examples. If I talk of pea plant has flower color either white or purple, right? So color of flower is one gene, and it might have two variations, white and purple. Understood? So that's inheritance. No, that's the alleles. Okay. So I hope the concept of alleles is a little clear to us. Okay. Moving ahead, if I see, we have homozygous. Now, what is this term homozygous? Homozygous. So, nucleus or a cell which has both the chromosomes with similar genes. Suppose I just gave you one example where one chromosome had black eye color from mother, one chromosome had blue eye color or brown eye color from the father. So these two alleles are different. But what if both these alleles are similar? Then what do I call this condition? I call this condition to be homozygous, right? Blue, blue, brown, brown, black, black. So it will be homozygous condition. As you can see here, TT, tall, gene for tall, tall, dwarf, dwarf. All right. Then you have heterozygous condition. All right. So now for heterozygous condition, you might have two chromosomes, one with one allele, the other one with a different allele. If the alleles are dissimilar, so it's a heterozygous condition. Okay. I hope I'm making sense to you, all of you. Okay. Let me show that to you and give you a little quiz. Okay, I'm drawing something on the board and you guess what. Okay. So these, suppose this is one allele, this is another allele. Okay. So it is homozygous or heterozygous? Yes, it's homozygous. So if I make one capital allele, another one smaller one, right? So it will be a heterozygous condition, correct? Yes. So it's heterozygous condition. Now coming back here, what are dominant traits and recessive traits? So before we begin, I hope you would be wondering, I'm just discussing these terms with you, but let me be clarified. Uh, unless we are clear with these terms, we will not be able to understand the chapter. So this has been taken from Bank of Biology. You can refer to the PPT there also. So one is a dominant trait and the other one is a recessive trait. So what is a dominant trait? I gave you one example earlier, that a child might have one allele which is from father and another allele which is from mother, right? But he will never have both these expressed. Suppose a child has black eye color from mother and blue eye color from father. Do you think the child will have one black and one blue eye? Of course not. right? So only one of the allele will be expressed, and the one which is more dominant, as we see in the society also, one which is more dominant will express over the other one, which is the weaker one, which is called the recessive trait, right? So if there are two alleles and one is able to mask the expression over the other, then it will be called dominant allele. If the other one, which is the weaker one, will be called recessive allele. Okay, then you have phenotype and genotype. Phenotype is what? Phenotype is what? Expression. Okay, so how you appear, what is your appearance, is your phenotype, physical appearance, right? And genotype is the genetic constitution. Okay, let me give you some examples. For example, um, hair color. ठीक है? If I talk of hair color or eye color, one might have blue eye color, one might have black eye color. Now, how does that matter? Okay, let me show that to you on the whiteboard. Let me clear this and then, okay. So, an individual can be homozygous or it can be heterozygous, right? अगर eye color की बात करें, so black, I'm making black like this, and blue eye color like this. Okay, so this individual is heterozygous, right? This individual is heterozygous, and it will not have one blue and one black eye color. So if this is blue, this is black, and if I say blue is dominant, so the child will have which color of eye? Blue eye color. So appearance of the eyes will be blue, right? So blue will be what? Phenotype. And this is the genetic constitution. So this will be the genotype. Okay, allow me to give you more examples. For example, if I talk of pea plant, so pea plant can be tall or it can be dwarf. If I express like this, these are the two alleles. Now we have understood this. Okay. So in heterozygous condition, it will be like capital T and small t. Okay. So what do you think? Which character will be expressed? Dominant or recessive? Yes, dominant. And dominant is the tall one. So how do you think this will appear? How do you think this pea plant will look like? It will be tall. So a pea plant will look tall. So tall will be its what? Phenotype or genotype? Correct. It's phenotype. Okay. Appearance, physical appearance will be phenotype. Correct. And if I say this is the genetic makeup, one capital T, one small t, so this will be the genotype of one individual. Okay. Okay, give me more examples. Geno, please forgive me for this. My style is, I'll work upon that. Okay, and if I show you more examples, one plant will be small t and small t. Now what? It has no dominant trait. So who will be expressing itself? Of course, the recessive one, right? So this recessive gene will be, how will it look? It will look dwarf, right? This plant will look dwarf, correct. So dwarfness, this plant appearing dwarf will be the phenotype, correct. And the genetic makeup, the genes which are available will make up its genotype. Wonderful. So this is, I hope, clear to everybody. Okay. Coming up next, moving on to the next page. Okay. So next will be hybrid. Hybrid. Okay, we will discuss this when, unlike parents meeting individual produced by two different individuals, we'll see that with the examples. So this person, we are not going to forget him. He's father of genetics, Gregor George Mendel. He'd been working on what? A quick revision. He'd been working on Pisum sativum. Correct. which is the garden pea plant. Okay, one more thing. One question often comes in exams: why only pea plant? Why did he only choose pea plant? He could have chosen people, banyan tree, anything, but why only pea plant? So there are certain reasons, important factors. So number one, it's true breeding. True breeding. Now, what do we mean by that? True breeding. Let me write that. It's true. Forgive me for this, please. Okay, it's true breeding. I'm not able to write properly. It's true breeding. So, what do we mean by true breeding? True breeding means this individual can only have either of the traits. I'll give you examples to explain this. For example, uh, this individual pea plant will either have tall plant or dwarf plant. It will never have intermediate height. ठीक है? This plant will have either either purple flower or white flower. Never a mixture of these. Okay, this plant will always have a yellow seed color or green seed color, never lemon yellow or greenish yellow. So it will either be green or yellow. So it's true breeding, right? Second is the short lifespan. Suppose, had this Gregor been working on banyan tree, do you think he could have studied more generations? Of course not. It lives for 100, 200 years. So it is difficult for the scientists to study different 10, 20, 30 generations of one plant. So short lifespan is another important factor. Short lifespan is another important factor because of which Gregor chose this. All right. Now coming back here, this pea plant has seven contrasting traits. Seven contrasting traits, which I will show you with the help of a table. Okay, this is the experiment. This is the experiment. This often comes in board examination also for 12th practical. This is the is artificial hybridization. So first step is emasculation, selection of two different plants. Then emasculation. Emasculation means removal of anthers, so that we dust our desired plant with desired pollens. Okay, and then you have pollination with the desired pollen, so as to prevent cross pollination. We will discuss this later. Okay, so these, as I told you, these are the seven contrasting traits that you see in a pea plant. It will be tall or dwarf. It will never be intermediate in height. Flower position, it will be axial and dominant. As you can see here, the flower position is axial or it can be dominant. Correct? So it will never be a mixture of this. Then you have flower color, as I already told you, it can be either violet or white. Pod shape, inflated and constricted. You must have seen the this pod of the Pisum sativum. मटर के छिलके देखे हैं हमने. Either they are inflated or they are constricted, wrinkled sort of. Okay, then you have pod color, green or yellow. Seed shape, we have read this in grade 10th also. Seed shape can be round or it's wrinkled. Then you have seed color. One very important thing that we all need to remember is the dominant color of the seed once they have matured is green. हम उल्टा कर देते हैं अक्सर. So we all need to be very careful. The dominant seed color is yellow and the recessive seed color is green. I'll keep asking you intermittently. All right. Coming ahead. Then you have monohybrid cross. You remember we did heredity in grade 10 also, and we had done these monohybrid crosses when we cross a tall plant and a dwarf plant. Um, so then we see the first generation, second generation coming up, I will explain this with the help of a worksheet. I will upload that on the channel, and then you can do that. All right. Suppose this is the tall plant, and that's the dwarf plant. So if we multiply cross this plant, so it will be separation of gametes, which is the second law of inheritance, and then the first F1 generation, which is also called the filial generation. Let me show that here. So if I cross one tall, this is homozygous tall, now we all are aware of it, and we cross it with homozygous dwarf. So if I'm crossing it, then it will first be separated into the gametes. So gametes will be capital T and gamete capital T. This is law of segregation. So this can be explained like one chromosome, other chromosome. So when gametes are formed, these chromosomes, sorry for poor drawing. So these chromosomes, they move on to different gametes. Okay. So are the genes also separated this way. So these genes, they get separated. This is law of segregation. Same ways we will see the dwarf genes also being separated, small t. Now we have a fair chance of um, fusion of any gamete with any other gamete. So I would suggest you all, instead of making cross, you can make a Punnett square like this and write all the gametes, all the genes combination alleles towards the edges, small t and small t. Now you can start filling in. So it will be like the matrix that we do in maths, capital T, small t, capital T, mm, capital T, and small t. What will come here? Yes, correct. Capital T and small t. Wonderful. Capital T and small t. So as you can see here, all of these, they are all capital T and small t. Now I request you all to please tell me if this is homozygous or heterozygous. This F1 generation, this is the parent generation that were crossed, and this is the F1 generation. Sorry, this is the F1 generation which is formed, right? So capital T, small t. So this will be correct. This is heterozygous. And how will it look like? What will be its phenotype? Tall. Wonderful. So this is tall. Correct. So this is the F1 generation, 100% tall, all tall. Now, what if I cross or self pollinate the F1 generation? We can write on selfing. Selfing means self pollination, not that phone wala selfing. Okay, so on selfing, that is F1 into F1 crossed with F1, right? So when these are multiplied, can you tell me the genotype of F1 generation? Correct. It's capital T, small t into, that's crossed with capital T, small t. So when these were crossed, I have told you earlier, first will be the separation of the gametes, law of segregation. Capital T, small t, capital T, small t. Wonderful. Now, again, the Punnett square that we're going to make here. Yes, correct. Capital T, small t, capital T, small t, capital T, capital T. What will we have here? Capital T, small t. Children, please don't misinterpret this for pi. It's double T, it's capital T and small t. Same ways here, small t and small t. Wonderful. So now I would request you all to help me know what are the phenotypes. You can see all these are different genotypes. So capital T is a genotype. How will it look like? Correct. It will look like it will be a tall plant. Wonderful. Both of these, both of these will be, it's heterozygous, but again, how will it look like? Tall. Correct. यानी कि एक कैपिटल टी था जीनोटाइप और दो क्या थे? कैपिटल टी स्मॉल टी. वंडरफुल. कैपिटल टी and small t. And the third, fourth one that you can see here. I'm running out of the battery. Allow me a time, a little pause. Um, and the third one will be small t, small t. It will be what? It will be a dwarf plant. Correct. Very good. Okay. So it will be what? Why am I not able to write? Okay. So this will be, there's some technical error here. I'm not able to write that. It's it's small t and small t, which is like, oh sorry, it will be how phenotype we are writing here. So it will be dwarf. It will be small t and small t. So if I ask you what is the genotypic ratio and what is the phenotypic ratio in the F2 generation, now we have known that the result of this F1 cross is filial 2 generation. We call it F2 generation. So F2 generation mein if we see, there are 3 tall and 1 dwarf. So phenotypic ratio will be, what will be the phenotypic ratio? 3 is to 1, right? 3 tall and 1 dwarf. Phenotypic ratio will be 3 is to 3 upon 1. 3 is to 1. And if I talk of this genotypic ratio, so genotypes, you can see we have 3 different type of genotypes. One is this capital T. One is this capital T small t, which is present in 2, and the third one is small t. So genotypic ratio will be 1 is to 2 is to 1. Correct? So genotypic ratio will be 1 is to 2 is to 1. Correct? So this is the genotypic ratio. I hope I could clear this to you. And we can be online meeting one day for the online class, clearing all the doubts and the queries that you have. Okay. So I before I end this session, I would want to give you one trial. I would want you all to now work upon. I would want you all to work upon another cross, um, which is a cross between capital T and capital T small t. So one is um, heterozygous and another one is homozygous. So now you do at home the cross between capital T and capital T small t, homozygous and heterozygous, and do share the results with me. You can write in the comments on the video what were your results in the F1 and F2 generation. That is, in F1 and F2 generation, what are the genotypic and the phenotypic ratios. So I request you all to be sharing that with me. Thank you so much for your kind cooperation. Thank you.